Okay, so this is lesson 1 for module 5. So module 5 is the first of the year 2 modules, and this will be examined on paper 1 next year. And it's basically a module that is about the sort of fundamental basics of some of the further biology. So we start off by looking at homeostasis. We then go on to look at respiration and photosynthesis, the nervous system's in there. So it's control, sort of coordination and energy, the overriding topics of this. So we're going to start off by looking at homeostasis, and we're going to look at the kidney as our first part of that we are going to be looking at. So today is just going to be a basic introduction to the physiology of the kidney. So we're going to have a look at the gross physiology and some of the sort of finer physiology of the kidney. So, but first of all, we're just going to cover what is excretion. So this is a recap from GCSE. So I'd like to have a think about this whilst I'm talking you through it, and we'll come back to it in a second. So what is the definition of excretion? So remember from GCSE, we've talked about this before. Excretion is not feces, but it is urine. So what is the difference between excretion? What substances are excreted from the body? So there is a difference between excretion and egestion. Egestion is when things pass straight through the body and nothing happens to them. So that's your feces. So what is excretion? What organs are primarily responsible for excretion? So there are a couple that we're going to be looking at, and obviously the clues in the title of the lesson for one of them. And why do things need to be excreted? Okay, so why do we need to get rid of these? So let's start with the first one. What is excretion? And it might be best if you wrote these down in your notes. So excretion is defined as the removal of metabolic wastes from the body. These wastes are the products of chemical reactions in the body. So as I said earlier, feces is not excreted, it is ingested. It's just the fibre, the non-digestible part of the food that you take, passing straight through your body and leaving. Okay. What type of things do we excrete? Carbon dioxide and urea are two of them. We do excrete others. We excrete water, that's a product of a chemical reaction. We excrete salts, they're also excreted. But carbon dioxide, big product from respiration. Urea, I will talk a little bit more about in a second. So what are the organs that are responsible? Well, it's the lungs for carbon dioxide and it's the kidneys for urea. And why? So we need to maintain a constant internal environment. That is what homeostasis is. You did that at GCSE. So we can't have things like pH, water content, ion content of the blood being altered too significantly, otherwise it can have a massive knock-on effect. So carbon dioxide, we need to excrete that, as it says there, it can interfere with the oxygen carrying capacity of the blood, and you have done a little bit about this with Mrs. Ford, or it can dissolve in the blood and form carbonic acid, which can alter the pH of the blood and interfere with cellular processes. We can't have too much urea in the blood because it's poisonous. We can't have too much water in the blood because that can affect the water potential of cells. We can't have too many salts because that can affect the pH. Okay, so all of these things need to be regulated. So let's have a talk about nitrogenous compounds. So you did this at GCSE, but I'm just going to remind you again. The body cannot store excess amino acids. So a high protein diet, you don't store the protein and use it for later. You can't store them. You break the protein down into amino acids, and then those amino acids are absorbed, and then they get transported to the liver. Now, when they're in the liver says, okay, we need this amino acid, we don't need this amino acid, and anything that are extra, they are deaminated. Now, if you remember your structure of your amino acid, you have your amino group at one end, your carboxylic acid group at the other. They remove the amino group, and you end up with this very toxic compound called ammonia. You should know the smell of ammonia. It's a sort of very pungent smell. You can often smell it, in urine. And basically, we can't have that. It's very soluble, very toxic. So it gets converted in the liver into something less toxic and less soluble, called urea, and that happens in a process called the ornithine cycle, which you will do with Mrs. Ford when you look at the structure of the liver. So ammonia is NH3, we add some carbon dioxide to it, and we end up with urea. So that's a chemical formula for urea there, and some water. And as I said, that happens in the liver. And then the liver, okay, secretes this urea into the bloodstream. It's transported to the kidney, where it then will be excreted. So urea is made from excess amino acids. Excess amino acids are deaminated, converted via ammonia into urea, and then urea is transported to the kidney. So this is the most important part of this process. So you should have this diagram or have access to it or have a look at it. And we're just going to very simply label the structure of excuse me, the urinary system. So as with all biological diagrams, we're going to sort of start off by a reference point, and we're going to start off by labelling the right kidney. Oh look, it's on the left. Remember, all biological diagrams are reversed. So the left is on the right and the right is on the left. We then got two large blood vessels. One bringing oxygenated blood down, which is why it's red, and one removing deoxygenated blood away, which is why it's blue. Please remember, blood really isn't really red and blue. So what is this large main blood vessel that's passing all the way down? That's coming from the heart, full of oxygenated blood. It is, of course, the aorta. So a branch of the aorta, we actually have two of these, we've only got one labelled, heads into both of the kidneys. So this is called the renal artery. So we have one going to the left kidney, one going to the right kidney. These are the renal arteries. So they take the oxygenated blood in. The blood then, this blood also contains the urea. The blood then comes back out after it's essentially been filtered by the kidneys. This is basically what the kidneys do. They filter the blood. But they will also have removed the oxygen, which tells you that something in the kidneys requires energy, because the oxygenated blood has become deoxygenated because some of it has been used for respiration and has released carbon dioxide into the bloodstream. So we then have, leaving the kidney, the renal vein. And again, there are two, one from the left, one from the right. So these then enter back into this major blood vessel here. This major blood vessel is the one that passes all the way up to the body, carrying the deoxygenated blood back to the heart and then to the lungs. So it is, of course, the vena cava. So that's our blood vessels. So we have two kidneys. They're located on the back wall of the abdominal cavity. And they then filter the blood, and they produce from that blood urine, which has to pass down these two tubes here to the bladder. So these are the ureter. So again, you have two of these, one from the left kidney, one from the right kidney. They are taking the urine down to the bladder, which is the organ below. Now, the bladder has at the bottom of it a sphincter. So a sphincter is a ring of muscle. That ring of muscle contracts. You don't have control over your sphincter from your bladder until you're, I don't know, 2 or 3, I think, when you're potty trained. And that sphincter then releases the blood out via the urethra to the outside of the body. Now, the urethra in females is much shorter than the urethra in males, because the male urethra travels all the way down to the penis. In females, the urethra only carries urine. So the female has, leaving the body, the urethra, the vagina, and the rectum and the anus, whereas males, their urethra, not only does it carry urine, it also carries sperm in the form of semen. So males only have, the urethra, and they have the rectum via the anus. Okay, right, so that is the urinary system. We are going to focus in now on the kidney. Now, if we were in school, we'd have done a dissection of a kidney, and hopefully we'll do one when we reconvene. So this is a section through a kidney. Okay, so we've just cut a kidney longitudinally, and we're looking at it through that. Now, we're going to sort of have a look and a little discussion. And again, you should have this diagram available to you, and then you can check it off at some point. So, on the very outside of the kidney is a fibrous capsule. And you can't really see it on here. It's a very sort of thin membrane-like structure. If you ever buy kidneys from the shop to make Slayton kidney pie, That fibrous capsule has been removed. It's not there because it's just quite tough and we don't want to eat with it, but it's there for protection. Then just inside we have a layer. So this is this layer here, this sort of paley brownish layer. You can sort of see it goes all the way around here. This is the cortex. So this layer here, this is called the cortex. This is the outermost layer of the kidney, and that is where a particular part of the functional unit of the kidney is situated, and we will talk about that a little bit later. Then we have these individual units here. These are called pyramids because they are three-dimensional structures. They are sort of pyramid, triangular in shape. And these make up this area here, which is called the medulla. So this area here is called the medulla. And again, it's where particular parts of the functional unit of the kidney are located. Slightly different in colour. We have these pyramids, and each one here, as you can see on this diagram here, has an apex, so sort of a point. And basically, the blood is filtered in this area. The bits we want to keep, I'll put back into the blood essentially in this area. And then what we want to get rid of makes its way down into this area here. Now this area here, just to cause confusion, is called the pelvis of the kidney. This white area here is called the pelvis of the kidney. So we've got the outside, the cortex, the medulla in the middle, the pelvis right there in this area here. And then we have various blood vessels. These are branches of various arteries and veins that you don't. And again, here we've got probably the renal vein coming in here, and then you've got the ureter down here. Now, the functional unit of the kidney is a structure here called a nephron. Now, you cannot see a nephron with the naked eye. This one has just been put on this diagram here to give you an idea of the location of the different parts of the nephron and where they are situated within the kidney. So you can't see the nephron. You have about 1 million nephrons per kidney, and we'll come back to those a little bit later. So these are some key parts of the kidney for you to jot down. So we've got the fibrous capsule on the outside for protection, the cortex on the outside. This contains these things called Bowman's capsules, convoluted tubules, and blood vessels. I'll make sense in a minute. The medulla, the darker area on the inside, where the loops of Henle are, the collecting ducts, and again more blood vessels. Pelvis collects the urine, and the ureter carries the urine down to the bladder. Okay, so again, the same diagram, but now we've got a slightly extra bit on the side. So here was our section through the kidney. And now we're looking at where this part here, the nephron, where it is located. So over here, and again, you wouldn't be able to see this with the naked eye, we have the two distinct sections. We have the cortex at the top and the medulla in the middle. These are our nephrons here. So our nephron, you have this large sort of capsule area here. Then you have these wiggly tubes. They go down into a loop. Then you have more wiggly tubes, and then they meet up in this thing here called a collecting duct. So in comes the blood. This bit filters the blood. This whole bit is where various bits of reabsorption happen. So we have to filter some things, then we have to keep some back. And then this bit is where all the urine collects. So you have about 1 million kidneys, about 1 million nephrons in each kidney, but lots and lots of nephrons collect to one collecting duct. So you don't have 1 million collecting ducts. You just have, quite a few joining into one, quite a few nephrons joining into one collecting duct. So let's have a look at the nephron. So the nephron is the functional unit of the kidney. There's about 1 million of those in each of your each kidney. So they're about 14 millimeters long, so not that long, and they sit in about in the cortex and the medulla. So everything. Let's see if I can make this work. So everything sort of here, in the top half, everything here is located in the cortex, and everything down here is located in the medulla. Okay, so we get rid of that. All right, so we have the cortex and the medulla. So we're just going to go through now and label each part of the kidney. So we're going to start, and we're going to sort of work our way around the diagram here. So we're going to start down here. So this is where blood arrives. This is a branch of the renal artery. Okay, has brings in the oxygenated blood that has come from the heart via the aorta. Okay, so the blood that's in here is going to be high in oxygen, it's going to be high in glucose, it's going to have plenty of urea in it, it's going to have all sorts of other odds and ends in it, and it comes here. That then starts to separate, and it separates out into arterioles. Arterioles are small arteries, and this one here is called your afferent arteriole. Okay, your afferent arteriole then separates up into a bundle of capillaries inside here, which make up something called the glomerulus. So your glomerulus is this bundle of capillaries that's all like a big knot of capillaries that sits inside something called the Bowman's capsule. We are going to do lots more about this later, so don't worry too much, but now I just want you to be able to label different parts of it. So we've got the glomerulus, which is a knot of capillaries, sat inside the Bowman's capsule, which is containing it. Then your afferent arteriole brought all those capillaries in. The capillaries then all join back together and form the efferent arteriole. So afferent goes in, efferent goes out. And you will notice, or you should notice, that our afferent arteriole is slightly wider than our efferent arteriole. This brings about an increase in pressure in the blood that's situated inside the glomerulus. And that is important, which we'll talk about again in the next lesson. So, the efferent arterioles then split into lots and lots of capillaries, which then form this mesh, this net, all over the rest of the nephron. And again, that is important. And again, we'll come on to that a little bit later. So that's the blood. They then join back up to form a branch of the renal vein. That then leaves the kidney, goes back to the heart, lungs, etc. So that's the blood system. Artery brings in the blood. Afferent arteriole splits, forms the glomerulus. The glomerulus then goes back together, and the efferent, and then they form this net of capillaries, and then they join back up together to form the renal vein. Now we're going to have a look at the nephron. So we've got the Bowman's capsule. The Bowman's capsule surrounds the glomerulus. When it's surrounded it, it then forms this curly tube here. It's called the proximal convoluted tubule, which translates as the first coiled tube. So it's a convoluted, so it's all wiggly. It's a tubule, it's a small tube, and it's the proximal, so it's the first. That then goes down into form the loop of Henle. This is the loop of Henle. It goes down, that's the descending limb, and it goes up, that's the ascending limb. So we have this loop of Henle. So it loops down, it loops round, it loops up. And then it forms something called the distal convoluted tubule. Okay, and then all of that joins up into the collecting duct down here. So Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct. Those are our parts of our nephron. Our nephron is our functional unit of the kidney. So essentially, we filter blood, we reabsorb things we want to keep, we reabsorb water, we reabsorb more things we want to keep, we've made urine. All right, and we're going to go over all of that in much more detail over the coming lessons. So these are some electron micrographs of varying different parts of the kidney. So we've got some different sections through different parts. So we're going to start off with these ones up here. These ones up here are sections, these are all sections of the cortex at the top here, so this upper portion of the nephron. So you can see, or hopefully you can identify, these are these glomerulus, these are the glomeruli, sorry, so this is a glomerulus, and this is a Bowman's capsule. How do we know it's a Bowman's capsule? Because it's a big space. So there's two in this diagram here. There's one in this diagram here. This is the glomerulus. This is all the blood vessels. And this is the Bowman's capsule, this space that sits all the way around it. This is the start of the proximal convoluted tubule. This is a distal convoluted tubule. You've got a million nephrons all sat together, all sat next to each other. They're all intertwined. You are not experts on the kidney. You are not expected to be able to spot your distal convoluted tubule from your proximal convoluted tubule from your collecting duct. But you are expected to be able to identify a Bowman's capsule. And you identify that because it's got this open space within it. If we look over here, on this diagram here, they have labelled the distal convoluted tubule and the proximal convoluted tubule. You can't really tell a lot of difference between them. All right, you would not be expected to. You might be asked to identify a tubule and you'd be given marks for identifying either of them because then you'd at least know the names. But you can see what they're made up of is cuboidal epithelial cells, so little cubes, right, all located round. They're quite, organised. They've got big nuclei in them, and again a bit of a space in between. This one's got some stuff inside of it. This one's empty. Depends on the particular part of the cycle it's in. Now we're going down to the medulla down here. So we're looking basically at loops of Henle, and we've got two different versions. We've got a transverse section and longitudinal section. Transverse section, we've got these nice circular shapes. Again, you are not expected to know whether you're looking at the ascending loop of Henle, the descending loop of Henle, or a collecting duct. But you are expected to be able to say that you are, identifying, you might be given what possibly could this be. So you could label that. This is a longitudinal section. So we've cut down through essentially like a cylinder. Of course, because they're not all located straight, you're not going to get, a beautiful cross section through. But we've got much longer open spaces as they've cut down through those loops of Henle there. Okay, so that's the sort of thing that we identify. So you need to be able to look at an electron micrograph of a kidney and identify, first of all, whether you're looking at the medulla or the cortex. The key difference is the location of the Bowman's capsule, and you'll then be able to, you will be expected to note which bits are the Bowman's capsule. So that's the end of this lesson today. All I would like you to take away from this is the labelling of the different sections of the kidney and the labelling of the different parts of the nephron. Okay, which part of the kidney is which part of the nephron is which. And then we will carry on with this next time.